Hello everyone, my name is Harpreet Singh, and today we'll be discussing about general principles of food microbiology for CFSO 2022. The whole idea here is to get short videos. This would be around 10 or 15 minutes. I do not know how long will it go, but the idea is to get short videos and so that your attention span is retained. I want to give a small introduction about myself before we dive deep into this topic. Yeah, this is me, and currently I'm a PhD scholar at NIT Rourkela. My project is on development of effluent-free food processing operations using non-ionizing electromagnetic radiations. There's a lot of words that I've said right now, so just understand that I'm working on a government-sponsored project where I have some industrial partners as well. And I was a professor at Vignans University. I have so far catered 200 or 250 students for CFSO. This is just a small information about myself to give you an idea that I'm a qualified person to give you the information or train you for CFSO. Anyhow, there are more to this about my qualification, but this video is not about me. This is about food microbiology. So, microorganisms, if we try to break down this term, we have two terms that are coming up. We have micro and then we have organisms. Micro is something which is small, and then we have organisms which are something living. So, microorganisms are your living organelles which are very small and cannot be seen with your naked eyes. And depending upon the type of microorganisms we have, they can either provide some positive implication on human health or they can provide some negative implications on human health. Then these microorganisms require certain set of conditions for its growth and survival, like conditions in terms of its temperature, conditions in terms of its pH, conditions in terms of its water activity. When these conditions are disturbed, the microorganisms tend to die or their growth is retarded. Now, common sources of microorganisms include your soil, water, air, and any sort of contamination which could possibly happen. These, uh, these will introduce microorganisms to our system, and then microorganisms can destroy it either by higher temperature, like in the case of your pasteurization. You are bringing your milk, you're boiling your milk. Boiling is a very layman term over here. I still use it. You are heating your milk to a certain time temperature combination, which is killing your microorganisms. Then you have alteration in terms of pH, which is again killing certain group of microorganisms. Then you're using certain set of chemicals like preservatives in your food that are killing your microorganisms, and at the same time they are retarding the growth of microorganisms. Now, if we see about the classification of microorganisms, we have algae, we have protozoa, we have molds, we have fungus, we have bacteria, we have archaea, we have viruses. This classification is taken from a research paper, and this is highly reliable. I mean, a very reputed journal. So you can rely upon this classification. There are many classifications that you will be seeing on internet. So for our discussion, we'll stick to this. For your exam, you stick to this. This has been taken from a research paper. I do not remember the name. It is from some nature general. Then we have factors affecting the growth of microorganisms. We have intrinsic factors and we have extrinsic factors. In intrinsic factors, we have water activity, we have pH, we have nutritional contents, we have antimicrobial agents. So your antimicrobial agents are like your preservatives in your food, which are preventing or which are hindering the growth of microorganisms. Then we have nutritional contents. Microorganisms do require certain set of nutritional for its growth. They require proteins, they require carbohydrates, they require fat, they require water. Water we'll be discussing in your water activity part. I'm just giving you a brief idea of what we want and what microorganisms expect for its growth. So they expect certain sources of energy as well. And coming to pH, they they require certain pH conditions which are favorable for its growth. Then we have water activity. Again, water activity is giving you an idea about available water in the food for or the free water which is available for the growth of microorganisms. So there are certain set of microorganisms which will be growing in certain water activity range, and there are certain set of microorganisms which won't be growing in that range of water activity. Then we have extrinsic factors. Extrinsic factors, anything other than composition of food, okay, is implicating or impacting the growth of microorganisms. We consider that as extrinsic factor. We have temperature, the temperature in which the food is kept, the temperature in which the food is stored is the one is considered as an extrinsic factor. I'll give you an example. Like you have prepared curry this morning and you store it outside. You did not keep it in the fridge. Your curry might get spoiled by the end of the day. All right. So what you do, you tend to keep it in the refrigerator so that the temperature is controlled. The growth of microorganisms could be prevented. Then you have gaseous environment. The gases which are present in the atmosphere is also implicating or impacting the growth of microorganisms over here. Like say for example, the kurkure or lace packet that you get in the market, they are injected with certain gases. These gases are inert in nature, and they are providing a cushioning effect. First thing is that, and second thing, it is also preventing or hindering the growth of certain set of microorganism, and also it is preventing rancidity of your food. Then you have relative humidity, then you have microbial interactions. Again, there might be certain microorganisms which are inherently present in the food, which are desired in the food. So these microorganisms which are inherently present in the food, they are hindering or they are not allowing the other set of microorganisms to grow. Like in the case of curd or yogurt, you already have certain microorganisms which are there in the food. Okay, these microorganisms which are there in the food, they are they are actually preventing the growth of other microbial microorganisms as well. Or you can take certain probiotic drinks as well. So if there's a microorganism which is trying to grow in in those conditions, the microorganisms which are already present in the food, they may hinder the growth of microorganisms which may cause or which may not be desirable in our scenario. And then we have microbial growth curve. So you can see over here there are different phases: lag phase, log phase, we have stationary phase, then we have death phase. Now we'll try to understand each phase over here. On your x-axis you have time, and on your y-axis then you have number of bacterial cells. Lag phase. In lag phase, what is happening? Your microorganisms are getting introduced to the food environment. Like you take an admission into the college, or you sign up for a new class. In the first step, you get accustomed, or you get to know your classmates, or you get to know the batch, or you get to know the class, you get to know your professor over there. Okay, that is an example of your lag phase. Then after certain time, this time, okay, you can see that there's an exponential growth in the number of microorganisms. The microorganisms are shooting up in an exponential number. Now this is your log phase. In this I mean your lag phase, the microorganisms understood, yeah, this is our environment, this are the resources that we have, this is the water activity, and chalo, these conditions are favorable for our growth, they will grow. Then in log phase, there's an exponential increase in the number of microbial cells. Then we have stationary phase. Now, here in the stationary phase, what is happening? The microorganisms that are actually born and the microorganisms that are killed or that are dead at a certain time, or a certain point of time, that is equal. There is no net increase in the number of microorganisms in the stationary phase. I mean, like if say for example, two spores or two cells are being introduced into the system, then two cells are being eliminated from the system. Then you have death phase. The term itself is quite self-explanatory over here. After a point of time, you can see that new cells are not being introduced into the system. only the cells which are existing that are declining in number. Okay, that is your death phase. So this is just whatever I've explained you. A small um, text is given over here. Lag phase, getting used to the environment. Log phase, a logarithmic increase in the number of microbes is seen. Stationary phase, no net increase in the number of microbes is seen. Then we have death. The rate of cell death is faster than regeneration. I hope this is clear to all of you. Then we have classification of bacteria. Now, classification of bacteria is quite tedious or quite complicated over here. Now, this classification is made of several factors. One of it is on the basis of nutrition or the mode of nutrition. Then we have optimum temperature growth. Then we have pH, optimum pH. Then we have salt required. Then we have on the basis of gases required. Then we have on the basis of morphology. Then we have on the basis of gram staining. And then we have on the basis of spores. So we'll see in terms of mode of nutrition. In mode of nutrition, we have phototrophs, chemotrophs, autotrophs, and heterotrophs. Phototrophs. So if you break down this term, photo and trophs, photo is something related to light, right? So phototrophs are those bacteria which gain energy from light. And again, the same thing over here. Chemotrophs, they are gaining their energy from chemical compounds. Then you have autotrophs. These uses carbon dioxide as sole source of carbon to prepare their own food. Then we have heterotrophs. These uses organic compound as a source of carbon. Then you have classification on the basis of optimum temperature. We have cyclophilic group, we have a mesophilic, then we have thermophilic. So for cyclophilic, the range is 0 to 20 degrees Celsius, mesophilic 20 to 50 degrees Celsius, and for thermophilic, we have 50 to 80 degrees Celsius. Now, different books and different resources will have different temperature ranges. More or less, the temperature range is same, and this I've taken it from a research paper. So, very reputed one, it could be reliable. Then you have optimum pH for growth. We have acidophilus, alkalophiles. Then we have neutrophiles. Acidophilus or acidophilus. Here this microorganisms or this bacteria grow when they have acidic pH. Self-explanatory in terms of name. Then we have alkalophiles. These are the one which grow in an alkalic pH or a pH which is greater than 7. Then we have neutrophiles which grow at a neutral pH. Then on the basis of salt requirement, we have halophiles. Then we have halotolerants. Halophiles. Philes over here indicates love. So one which love salt, they require high concentration of NaCl. Then we have halotolerant. Again, it is tolerating something, right? So halotolerant do not require NaCl at the first place, but they can tolerate. Since in the name we have tolerant over here, they can tolerate low concentration of NaCl over here. Then we have on the basis of gaseous requirement. First, we have obligate aerobes, facultative anaerobes, aerotolerant anaerobes, microaerophiles, obligate anaerobes, and capnophiles. Then, first thing I want you to understand is the terminology over here. Okay, aerobes require oxygen and cannot grow in the absence of oxygen. Here, the term I want you guys to pay attention to is obligate. They are obligated to grow in terms of aerobic environment. Okay. Here again, one more similar term you are able to see, that is obligate anaerobes. So they are obligated to grow in an atmosphere where there is no oxygen. Okay. Obligate aerobes and anaerobes. I believe that is clear to you. Then you have facultative anaerobes. Facultative anaerobes do not require O2. Okay. And then you have aerotolerant anaerobes. Here again, in like your previous case, Halotolerant. Again, the term tolerant has come over here. Someone or something is being tolerated over here. Okay, uh, aerotolerant, anaerobes. So do not require O2. Anaerobes, uh, clear over here. They do not require O2. I believe that is clear now. Aerotolerant. Now tolerant is also there, but for growth, but can tolerate the presence of O2. I mean, like at if the concentration of O2 is very low, they can tolerate the presence of O2. Then we have microaerophiles. So low concentration of O2 or oxygen is required. Then we have capnophiles, require carbon dioxide for growth. I believe this slide is very clear to you. Then on the basis of morphology, we have spherical, we have rod shape, we have spiral, we have other shape, other. These are certain shapes that you can see over here. For spherical, we have monococcus, dicoccus, we have streptococcus, staphylococcus, all these are there. So monococcus, only a single spherical structure. Dicoccus, you have two spherical structures. Streptococcus, you have a strand of coccus structure. You have staphylococcus, an arrangement which is something similar to grapes. And if you the rod shape is known as bacillus. Again, in bacillus, two structure I want you to pay a particular attention over here. We have a structure called Clostridium. From here, you get Clostridium botulinum. Then you have Escherichia. Okay, from here you get Escherichia. Okay, or Escherichia coli, E. coli. This is the morphological structure of E. coli. I mean, the morphological structure comes from here. Then we have Vibrio. You must have heard the term Vibrio cholera. It comes from here. Then we have, on the basis of gram staining, we have gram positive bacteria. Then we have gram negative bacteria. In your gram positive bacteria, you have only a layer of peptidoglycans. Okay, and whereas in your gram negative bacteria, you have a layer of peptidoglycans plus an outer layer. Then, on the basis of spore, we have endospores, we have exospores, endospores within the cell, and exospores outside the bacterial cell. Then we have non-spore forming bacteria. All right, guys, that is it for today's lecture, and I'll see you in the next one. If you like this video, please make sure to like and consider subscribing to my channel, and also please follow me on my Instagram channel.